A warning. This episode contains graphic descriptions of gun violence and violence against women and children.
A LiSTNR production.
Semen is the crack cocaine of the fly world. And while maggots love a corpse, flies feast on live human flesh and fluids. So, could a fly frame you for murder? And how do police differentiate fly artefacts from human biological material at a crime scene? In this week's Rewind episode, former host Kathryn Fox talks with forensic biologist Dr. Annalisa Durdle about redefining what it means to be a fly on the wall.
I was actually doing my master's degree at Auckland University in New Zealand, and as part of that I actually just had a job, as you do as a master's student, where I was cleaning the labs. So these were the labs that evidence from crime scenes were examined in, and it was a great job. You got to wash floors, clean blood off stepping plates, all that delightful thing.
Very glamorous.
Yeah, it's very CSI. So basically, I would go in there in the morning and you'd see the odd dead insect around, and it just got me thinking, is it possible for flies or insects to actually transfer DNA between surfaces themselves? And the more I kind of explored that area, there had been some work in Germany about fly poo, and they had kind of identified that if you have a lot of fly poo at a crime scene, it looks very similar to the sort of bloodstain patterns that you will get from something like blunt force trauma, or even something where there's higher force involved, like a gunshot. So this raises the question, if somebody doesn't have that training to be able to distinguish between fly poo or, kind of the genuine biological material, is it possible that if they were to swab that, thinking it was potentially the blood of an offender, could you get their DNA profile or the person's DNA profile from that and potentially either identify an offender, or, of course, that can potentially lead to a miscarriage of justice if it contained the DNA of somebody completely unrelated to the crime that had taken place.
Having not seen what I could recognise as a whole lot of fly poo in my life on Earth so far, to mistake it, are we talking one or two small little deposits that can be mistaken for blood spatter or dried blood, or at a scene in which bodies are decomposing, are the number of flies that voluminous that you will actually get a fair bit of deposits on walls or on floors or wherever?
Yeah, so it really will depend on the circumstances, how many flies have access to the scene and things like that. But there are cases where, the apartment's been closed, and the flies have entered in potentially through the crack under the door, and there has been sufficient fly poo or fly markings on the walls and things like that, that the people have walked in and actually immediately thought, what's been happening in all the rooms of this house. But what can also be interesting is if you've got a bloodletting scene, what the analyst or the crime scene investigators might be looking for is that blood stain that's an anomaly. So it's not one that doesn't fit in with the pattern with the blood that's likely to have come from the victim. And it's not unusual for people who have broken into houses or used knives in an attack to actually cut themselves. So there might be a drop of their blood somewhere else. So it's not part of a large pattern, but they may have the odd drop of their own blood around. And that could be the anomaly that investigators are looking for and may want to actually sample for DNA as well. So it can be fly poo en masse, or it can be one or two drops, depending on what the context is and what is of interest.
So the crime scene people are looking for pattern recognition, but also anything that falls outside that pattern.
Yeah, you could say that, because a lot of what the bloodstain pattern analysts are looking for is not only the pattern to reconstruct what might have happened, but what are some really good target drops that we can use to actually maybe inform us as to who else might have bled at the scene or who has actually formed that drop.
So what can fly excrement, poo, actually tell you? What do they reveal? I mean, we think in human excrement, there's possibly DNA cells from skin that's lost during the gut. I have no idea what the digestive process of a fly is. And so what on earth they will be depositing? And are they actually drawn to blood to consume and deposit?
Right. So the very interesting thing, as part of my research, what I did is I actually offered them a number of food sources. So this wasn't just biological material, but it was also the sort of food sources you might find in a house normally. So pet food, for example, or you might have honey or tuna or things like this that might be at the house. It was kind of like a preferential study, If you had a great lot of pet food, would flies go for that rather than any blood source or human tissue, which I couldn't study the human tissue for ethical reasons, but certainly used human biological material for the rest. And so basically flies have a proboscis, so they uncurl that in order to eat, and it's got lobes at the bottom. So if you've got a liquid food source, they'll suck that up. But if you've got a dried food source or semi-dried food source, they've actually got these little teeth at the end of the lobes, at the end of their proboscis, and they can kind of scrape at things in order to eat it. They'll take that into their crop. They will then actually regurgitate that. So you do have fly vomit on surfaces, but the flies tend to eat that again. They'll eat that up. It's not 100% clear why they do it, but it's believed that they might leave it out there to kind of evaporate off the water. It concentrates it more and it gives time for the enzymes to kind of break down the food, and so they can digest it better. So that's kind of their process, and then it will go through their gut and get excreted out the other end. In some cases, it can really be well digested inside the digestive system. In other cases, it might pass through almost intact. So if you've got a fly that's feeding on biological material, it might actually pass, essentially, that human biological material quite intact through the system. And so what you're ending out with is kind of essentially still that biological material. But in terms of are they attracted to it? Interestingly, yes, they are. They are attracted to it, and predominantly it's as a protein source, which female flies need to kickstart their reproductive system. But fun fact, they are most attracted to semen over all the biological materials, despite the fact it gets them drunk, it gives them diarrhea, and it kills them.
That is an awful lot to unpack. And given that a forensic biologist is not necessarily a behavioural expert in flies, how do you know that a fly is drunk in the first place?
I use the term drunk, and it might not be the correct technical term, but as part of my research, I recorded hours and hours, about 140 hours of footage of flies eating, pooing, vomiting, in order to see that kind of behaviour. And with the semen, what I noticed with flies is they basically staggered around. They couldn't walk properly, probably a bit like some people at 2 a.m. on a Saturday night in Melbourne. And it partially paralysed them too. So if you can imagine when you get a fly sometimes it's cleaning its forelegs, you'll see that it's kind of lifted those up and are running them together. I had this one particular fly that every time it tried to do that, it fell over. And in the end, it just sat down on its bum in order to be able to do that. So I use the term drunk, but it's really just it's losing its motor responses and things like that as well. But it'll go straight back for the semen. I call it the crack cocaine of the fly world.
If they're then regurgitating and then diarrhea processing, you assume it's not going to be very processed. If they've got diarrhea, it's going straight through them.
Well, yeah, it's not in all cases, but in some cases it'll be they blow a bubble out their bum rather than doing a nice poo. But it's not in all cases, but there's obviously something going on digestively as well.
What is that going to help you with at a crime scene, knowing that and having discovered those facts?
I think the interesting thing is that we really associate flies with decomposing bodies or with blood, and there's no end of literature that shows that flies are primarily through their olfactory senses are attracted to the smells of a decomposing body and things like that as well. But this kind of extends out what we know about what flies can be attracted to, and particularly at a crime scene, and the fact that we can get the human biological material. Like I say, you could use this as a target for a source of DNA as an offender. Flies move around at a crime scene, so they may carry the DNA's offender to another room where they deposit it there, and the offender is only cleaning up in the room in which they were actually committing the crime. So you've got that potential. The other issue is that it could throw a spanner in the works. So I know you've had previous guests that have talked about the sensitivity of DNA techniques and the amount that we can detect now that we might not have done previously. And so if you have these sort of fly artefacts that contain human biological material just on surfaces normally, and certainly in the instances of when you feed them semen or saliva, sometimes that fly poo can be completely clear. So you might not see it or you might not notice it. You have the potential for collecting these other sources of DNA, and semen in particular is a very rich source of DNA. And so you may have people that are turning up in these DNA profiles that had nothing to do with the crime scene, but their DNA has been inadvertently collected from the crime scene, and only because the flies act as a vector of their DNA and brought it in there.
Like pets, we've talked about pets in the past too, doing that, transferring DNA from someone whose hairs on them or who've rubbed their tummy or whatever, and they've come through and altered the DNA profile at a crime scene.
Yes. And I think what I found really interesting from my research is I actually had flies deposit their poo, and I actually didn't test that poo for over 2 years, and we were still getting full human DNA profiles from that fly poo that had been just sitting at room temperature for 2 years. So not in a fridge, not in a freezer. I had it in an envelope in a drawer in a lab at room temperature, and you were still able to get, not in all cases, but certainly in most cases where it was semen and a large number of cases where it was blood stains, you were still able to get those full human DNA profiles. And this was conducted well. We've developed the sensitivities, and it's become increasingly sensitive even since then.
2 years later, obviously it's not been exposed to weather and other flies breeding and consuming and regurgitating. Do you have any idea how many cycles that can go through before their DNA becomes undetectable? Is there any idea?
So I just want to clarify kind of your question. So you're asking that if a fly was to deposit poo, are the flies coming along, eating that, and then depositing it and that? No, I think that's a really interesting thought. However, from the research I did and watching all those hours of videos, like a really good reality TV show, they will vomit and then they will basically eat their own vomit again. It's part of their almost a digestive process. And while they will, on occasions, indulge in eating another fly's poo, it doesn't actually happen very often. So basically flies will go to their food source to get that protein, get carbohydrates or whatever they need. Then they may move away as well, because it's really about the purpose of having a body is not only to have that, be able to eat that protein, but also maybe find a protein-rich or moist environment for them to lay their eggs, so the maggots actually have a nice buffet when they hatch. So in that sense, it's not like they would necessarily do that. The only time I have seen that is when there hasn't been sufficient food for them. So they've run out of that resource, and then the only food available basically has been the other fly poo, and that's when they've eaten it. So if they can go away and get another resource, it is unlikely to happen.
If your research has shown that consumed semen then turned into poo by flies can actually reveal DNA, if you have a sexual assault victim who's been killed, is that theoretically likely to help you or practically help you find the potential offender through the offender's DNA?
There's a potential for it. And just to clarify it also, you can get DNA when they're fed on blood and when they're fed on saliva as well. So you can still get that from the other biological materials as well. In terms of whether you could do that again would depend on, I'm just kind of thinking of things that might throw a spanner in the works there. If they have fed on the semen and deposited it, sure. Are they also eating part of the victim itself, picking up the victim's DNA from skin cells and things like that as well, which might add a bit of a complicating factor. Flies will tend to go to orifices when there's a dead body. So that is generally your nose, eyes, mouth, vagina, but also if there's stab wounds or something, that might open up access to the body as well. So, this would kind of have to be almost a preferential orifice, and also to and eat if it's actually been deposited inside the body as well. But there is that potential, and I have done other research actually where a student did the research where we're kind of looking at, how does decomposition fluids and insect excretions and secretions actually affect the DNA that you can recover on the body? Does it degrade it? And we found actually no, it doesn't, to the extent that you might imagine, and that can still be useful too. So yeah, all potentially could be a useful resource, I guess.
Where do you get your actual semen samples for your research?
So that's a very good question, and obviously there's ethical requirements around that, because it's not only human biological material, but it's also the DNA and the profile that's going to be generated is very, that's very confidential information and very private. So we do have to get ethics approval to do that, but I do have volunteers. Once you approach people, they do volunteer. When I was first having volunteers, it was when I first started this research, I had neighbours that were kind enough to kind of donate. So my flatmate I was living with at the time got used to neighbours knocking on the door, handing home a sample jar saying, "Can you put this in the freezer for Annalisa, please?" They would keep it in their own freezer until they handed it over. I had another volunteer who would keep it in the frozen peas in his own freezer, because he knew the other members of his family didn't like peas. So that was where he kept it hidden. So I have been very grateful for my donors over the years, because it can be a rather awkward thing to request.
Coming up, Annalisa explains how examining bloodstains can help experts find evidence, leading to successful convictions.
How has this work been used in reality, in actual cases?
There was one that occurred in Nebraska in the United States, and in that instance there was a third-level apartment. Two victims were found. One was in the kitchen, one was in the lounge room, and both of them had a gunshot wound to the head. They also had gunshot wounds to the torso. And when the investigators arrived at the scene, there was certainly a lot of blood spatter that you would associate with gunshot attacks, but there was also a lot of other spatter that you might not associate so much with gunshots. So depending on the force that's applied to the blood source, depends kind of on how the pattern evolves, and also the size of the stains that are generated. So gunshot, you're kind of going to have stains, the majority of them will be less than 1 millimetre in diameter. If you've got blunt force trauma, so someone potentially has been hit by a bat, for example, then you're going to have blood stains that are more around 4 millimetres in diameter, or around that size. So when they got to the scene, they could see a lot of these larger stains, and not only were they present in the kitchen and lounge, they were also present in like the bathroom and the bedroom and things like that as well. So you've now got potential, if you don't have the training and recognise that as fly poo or fly artefacts, then you've actually, did something else happen here? Was there an attack? Was there a beating? If the victims that are there don't show any wounds associated with that, was there somebody else who's been beaten? Was there a fight and a struggle, and they've actually attacked the offenders as well? So you could further down the track do DNA analysis, but from the get-go and you're trying to interpret the scene, that can really throw some confusion in there. And that's what happened in this case. So the investigators contacted the experts to come in, and they were able to look at various factors. So when you have a lot of fly poo en masse, it's much easier to distinguish from the original biological material. So you have things like if you have a blood pattern generated from force on a blood source, you'll have this certain consistent directionality, because the blood will break up and all the drops will spray out in a pattern. But you can use that. They'll generally, if they've been airborne, they will hit a surface, but they also have a tail, and that's a bit confusing because the tail actually points in the direction that the blood was moving at the time it hit the surface. So you can actually basically draw a line essentially through the middle of a whole selection of stains, and they will, if you draw those lines, they will meet at a point, a congruent point, which will be the point of convergence, which is basically where the blood source was. If you're looking at fly poo, it often has tails as well, because the fly might be walking along, dragging its bum along as it poos, and so it ends out with these that look like tails of blood spatter, but they won't have that consistent directionality. So if you try and come into a point of convergence, you're not actually going to be able to by tracking their tails. So this is what the people did at this case. They looked, can we find directionality? Also, they found that fly poo with tails had proportionately longer tails in relation to the body than you would with normal blood spatter. So in that sense, they were able to say, no, we genuinely believe what's in the bedroom, what's in a bathroom is likely to be fly poo, and the main areas of attack were indeed the kitchen and the lounge where the victims were found.
How long roughly after they believe the victims were murdered did they arrive on the scene?
It would appear that it had been some time. The bodies were in a state of active decay. Obviously that doesn't happen quickly. This was the case where there was no windows open, so they did surmise that the insects had come in under the door, in the opening underneath the door. But given the temperature, I believe, in the apartment at the time was about 30 degrees Celsius. So that's pretty warm. That would increase the rate of decomposition, but you would surmise that there had been some time had passed between the killings and by the time the investigators arrived.
I'm just sort of trying to determine timelines, because obviously straight away you're not going to get fly poo on walls and fly deposits. It's got to obviously take a cycle or two of a fly's breeding in the decaying process.
No. So what will happen is that the adult flies will arrive at the body, and they're the ones that are feeding and defecating. So that can happen pretty quickly, and they may defecate within an hour or so of actually having fed on food, if not quicker. And so in that sense, that it's not taking long for that to happen. And even when I've been doing this videoing of fly poo, it's you're starting to get fly poo covering the container relatively quickly and in high numbers.
There was another case that you mentioned.
Yes, this was a case that was actually occurred in Germany, and in this instance, a mother and child were killed in their home, and they were actually able to determine that they had been killed 6 hours prior to being found, and I believe that would have been because there was another child in the house who was upstairs and unhurt. I think had been asleep at the time of the attack. And this was a very different, there was a different reason very much for them requesting the scientist in this case. And basically the woman and child had both been found in the lounge room, and there was, bloodstain pattern aligned. They were stabbed, that aligned with that. And there was also a lamp that hung from the ceiling, and it was approximately halfway between the two bodies that were located, and it was about 1.8 metres from the ground level. And there were some small stains found on this lampshade around the full circumference of the lampshade. One of the reasons that they wanted someone to determine if this was actually a fly artefacts or not is they had the identity of the killer, and the defence wanted to be able to say, look, my client was out of his mind at the time of the attack, and the way they wanted to prove that is they wanted to show that really brute force had been used in the killing of the child. And the pathologist had indicated that the knife had wedged at some point in one of the vertebrae of the child. And so the question was, were these stains fly poo, or were they small drops that had been flung off from the knife as the offender had jerked it out to remove it from the bone? Now, I think this is a really good demonstration of how it can be really difficult to tell when you only have kind of single drops and not, drops or fly poo, only one or two or a few rather than en masse. And these ones didn't have tails. They were circular, and that's also what can occur with general fly bloodstain patterns. In this instant, because they were so similar between what you would actually see for a bloodstain and what you'd actually also see with fly artefacts, they weren't really able to determine categorically by the morphology which they were. So what the scientists did, they used a bit of context, and they said it's winter. In this instance, the window had been open, and it was a clean environment. So they didn't note any rotting organic material, rubbish, pet food, anything like that. And they thought that it was unlikely that flies would have entered that house and actually deposited artefacts under those conditions. And there is the belief that you will find fly poo generally in warm areas where there's light, generally in higher up areas. I did do some research. We had access to a small house, just covered all the walls with butcher paper, sealed up all cracks with masking tape because those buggers will get anywhere, and had a pool of blood and released, hundreds of flies, and left them for a few days and came back and actually saw where the artefacts were in the house under different conditions. So in one instance, we had the blood and also water in a nice, well-lit room, and the rest of the house wasn't, all the windows were covered, and so you didn't have that sort of light. And they just stayed there, and their artefacts were found where you kind of would anticipate. It was around the food source, it was, on and around the window and on the ceiling, particularly in the corners. But when you start to change the conditions a bit, so if you've got a food source at one end, but there's light at the other end of the house and not in the room where the food source is, there'll be a lot of artefacts around the food source, and then there'll be a lot of artefacts at the other end of the house around the window, because they've gone for that. They've had their meal, and now they've gone for some warmth and some light. And these ones were conducted where it was a warm environment. You're looking at about 30 degrees outside. When we repeated that one where it was a cold environment and the food was in a dark room, but there was light at the other end of the house, the flies really tried to get down to that light, but they're very lethargic. Blowflies are very temperature dependent. Certain species will be active in certain temperatures, and this was obviously at the very low range for this particular species. So they're trying their best to get down the other end. Not many of them get down there. There's way fewer artefacts, and also most of them are lower down and not on the windows and things. So it really, again, is environment and context dependent on where you might actually find the artefacts.
Is there anything that can actually help differentiate fly deposits from blood?
It's actually very interesting, yeah. So basically the tests that you do, you have two types. The first one's presumptive. So that's that quick and easy kind of test that will have, false positives, but it tends to rule out something that won't be that biological material of interest. And then you can go on and for the stains for which they are positive, you can go on and do further testing, which is more specific to humans, and very much they are just immunoassays. So everybody knows a COVID rat. I'm sure everyone's taken one of those. They're also available to test for forensic biological materials. Those are specific ones for human blood, human semen, human saliva. So they do have the odd false positive, and particularly with higher primates and animals like that. And more research is finding kind of some more false positives, but that's kind of the process that we go through. So I did research with a large number of presumptive, and then these other, I refer to them as secondary, but also can be referred to as confirmatory tests. And it was interesting because it really depended on how old the fly poo was. So I tested some that were 3 days old, the fly poo, and basically it was possible for these tests to, particularly the immunoassays, rats for lack of a better term, they could actually distinguish between the two. So fly poo wouldn't give a positive result with those. But if you're looking at a 2-week-old fly poo, then interestingly, they weren't actually able to always make that distinction. And so you were sometimes getting positive results with those immunoassay tests when the fly poo was older. So I think this area is probably very nuanced and not really clear cut about what you can and cannot do to distinguish them.
So when you do your studies, like inside that house where you've put paper on windows and things, what's your toolkit when you go and check the house? What do you take with you?
Not fly spray. First thing I do, I go in there and I open the windows and go, get out of here. Because sometimes I had, hundreds of flies. It was just revolting.
What are you wearing when you go and do it too? What are you wearing to protect yourself?
Well, less so protecting myself in this instance because or certainly in this instance, I wasn't interested in necessarily testing for DNA. So I didn't have to wear any personal protective equipment that you would necessarily see at a crime scene, for example. The only weapons I had was my hands to shoo them away. But yeah, you'll open a window and they'll get out there pretty quickly. Certainly if there had been a DNA component to it, all of my research that has a DNA component, I'm wearing the full PPE. So that is a disposable gown, it's disposable gloves, hair nets, masks, booties if required, and certainly everything that you take into the laboratory or wherever you're doing your studies, everything is sterile, cleaned, DNA free. Because like I mentioned earlier, the levels of DNA that you can detect now are just so low that you just want to be minimising any of that potential for getting those mixed samples.
You are also renowned, either famously or infamously, for appearing on the Hamish and Andy show. And you were asked a question. Can I just preface this by saying this is not my question? But you were asked this. Can you get DNA from a fart at a crime scene?
It's a very interesting question, and I suspect it's one you would have always wanted to ask, Kathy.
Can I preface this again with no, I have never had any curiosity. And if you asked me off the top of my head, I would say no, you can't get DNA. But prove me wrong. I'm happy to be proven wrong on anything.
Well, actually, my answer back then will have changed based on very recent research. So fecal matter is not a best source of DNA, mainly because it's a bit of a hostile environment, shall we say. And also there's not a lot of DNA. It's basically the DNA is going to come from kind of the skin cells that the faeces passes through on the way out. And so certainly from a fart, I believe my answer at the time that if you had a hot burning curry, had farted, blown a hole through your jeans and actually deposited some fecal matter, even then it would only be a very outside chance of recovering any DNA. However, there has been very recent research that's come out where people are actually finding they're able to recover DNA from people from the air. And even they are testing surfaces where someone is sitting and, they're not contacting parts of that surface and they're still recovering DNA. So the understanding or the implication is that maybe it's passing through the air, and airflow would certainly help that. So I think now I might have to retract that, and if there was sufficient airflow, you never know what you might find.
I'm not sure if I'm glad or not that you've proven me wrong, because I don't know that I've wanted to really know that. But thank you. It's interesting that it's still a work in progress. We can tell Hamish and Andy that.
Absolutely.
Annalisa, thank you so much for joining us and explaining the importance of some of the research that people may just dismiss as unpleasant to the ear or unpleasant to the eye. And I know people aren't fans of maggots, but flies are actually quite a valuable source of information at crime scenes, which you've just demonstrated perfectly. So thank you very, very much for the work that you do and continue to do.
Oh, thanks, Kathy, and thanks for letting me share the fly poo love. I really appreciate it.
That's all we have time for in this Rewind episode. If you know someone who loves true crime, recommend our series to them. And check out our videos by searching LiSTNR Crime on TikTok or Instagram. I'm Dan Mullins. Thanks for listening.